Hello, it's Jane Blazeby here. I'm talking to you from just outside Bristol, from Backwell in North Somerset, and it's my pleasure to talk to you about the challenges of surgical trials. I'm going to share my screen now. I've been involved with evaluating surgical interventions for many years, but what's interesting is that the whole of the time from when I came to Bristol as a first-year medical student until I'd been a consultant surgeon for 5 years, during that time, which was 1982 through to 2005, I never saw a surgeon randomize a patient into a trial. I never saw a surgeon participate in a multi-center study. So I have learned a lot about trials, but very much in the last 10 or 15 years. So not a single patient during all of that time. And in the middle of that time, in the 1990s, I did a higher degree. And it was that time that Richard Horton published this letter in the Lancet, which has been very highly cited. And he said, surgical research or comic opera, lots of questions, but very few answers. And the reason why he said that surgical research was like comic opera was threefold. He said there are very few randomized studies published in the literature. He said the culture of surgical research is a problem, and the few trials that are published are of very poor quality. At that time, in the 1990s, only 7% of the papers in the published surgical literature were of an RCT. The most common papers were case series, just a single center, single surgeon. I've done 10 of these. This is how these patients fared. They didn't even report consecutive patients, and the outcomes were usually fairly short-term in-hospital complications. And then there's the issue of culture. And what has happened in the last 15 years is that this has really, really changed. And the main reason for it is that the Royal College of Surgeons have led on initiative in clinical trials. It's been amazing. And about in the 2013, I think the first centre was set up, the Royal College appointed initially about 5 surgical trial centres, then it became 7, and it's more than that now. They're listed here. And they were seed funded with relatively small amounts of funding, and they identified surgical speciality leads, so people that could champion trials in those fields. And they linked the trial centres with the leads, and all of those were linked to the multidisciplinary teams and particularly to trainees. And this initiative, which has absolutely changed the culture of surgical research, has meant that the number of surgical trials has massively increased. You can see that over that time period, there are a lot of new chief investigators in surgery and principal investigators, and over 50,000 patients recruited into surgical trials. It's really exciting to have been part of it. So the culture of surgical research is absolutely shifting. And what was once a very combative arena, single center, single surgeon, very much an individual, prestigious approach to research. So it was eminence-based, not evidence-based, maybe something like this. That is what has changed. And collaboration is the new competition. And trainees, surgical trainees, have led this, in addition to that initiative led by the Royal College of Surgeons. And what's happened is that the trainees were fed up with being asked to publish those single centre case series, and they realised that they weren't really worth the paper that they were written on. And they decided that there would be strength in collaboration, and they would be able to do good studies by working together to get large numbers of patients into national projects. And I just want to give you an example of a trial that I've been involved with, which has involved surgical trainees. It's called the Sunflower Study. And I mean, just to say that at the moment there are 16 of these different groups of trainees across the country all listed here. Then the first one was in Birmingham and they've spread right across the country. And in the Sunflower study, we have got 50 UK surgical centres recruiting patients. That's 50 is a huge number of surgical centres to be working together. And over 100 trainees, all of whom are GCP trained, are involved in it. And 34 of those trainees are what are called associate PIs, and I'll come back to that in a minute. And in Sunflower, we have an incredibly ambitious sample size to target. And Sunflower is aiming to recruit 13,000 patients. It'll be the largest surgical trial in the world, I believe. And in our internal pilot phase, which was scheduled just for 12 months, in order to reach our full sample over 4 years, our original internal pilot target was just over 2,000 patients, way bigger than a lot of surgical trials. And working with the 50 centres, working with the trainees and with our brilliant trials unit in Bristol, we, after 12 months, opened it last year, we recruited just under 2,000 patients. So about March 16th, just as we went into lockdown, we hit our internal pilot target and we got the go-ahead. Clearly, we're into a new challenge of how on earth we're going to complete the study. And trainees have been integral to that, working together to recruit patients. And a lot of them are what are called an associate PI. And the NIHR runs this scheme. I'd encourage you to involve your trainees in any of your trials where trainees can work with a consultant PI to be officially called a PI for their centre if it's registered and they complete the checklist. And that gives them some kudos and as well as responsibilities to really work on the study. And it's fantastic training. It's a fantastic apprenticeship to get fully involved in trials subsequently when they become a consultant. Now, if we have challenging surgical trials that are really difficult to recruit into, surgery versus no surgery, in Bristol, many of you know Jenny Donovan's group have pioneered the Quintet recruitment intervention, which aims to optimise recruitment and inform consent by using qualitative methods and looking in detail at screening logs. And the surgical trials we've led from Bristol and across the country using these methods that Jenny has really led on, we have conducted and completed successfully over 20 surgical trials. Trials that were said to be impossible to do have been made possible. It's been a real privilege working with those. I'm not going to talk about that in detail now. Now, the other reason why Richard Horton slated surgical trials was because of their design and their conduct. And I'm not going to talk about analysis because I believe if you get the design and the conduct of a trial right, then there's no need to have complicated analyses undertaken. And there are big challenges in surgical trials with bias. And one of them is around blinding participants, and another one that I'll talk about today is around blinding staff. And people often say it's just not possible to blind a patient. And we have been doing this in a surgical trial called Romeo. And we really tried it in the pilot phase. And in the pilot phase, it's a good opportunity to experiment with these methods and to establish whether it's possible to blind patients. And Romeo was looking at keyhole surgery. You can see here a picture of keyhole surgery versus a standard open cut. And we established in the pilot phase whether it was possible to blind the participants to whether they had keyhole surgery, a small cut, or a big cut. And then you can imagine everyone thought, of course, they'll be able to know whether they've had a big incision or a small incision. So what we did was at the end of the operation, this is an abdomen, a picture of an abdomen, whether the patient would have been randomized to keyhole surgery or to a major incision, they all had the same dressings put on them, which might have been like this, or it might have been a big dressing right across the whole abdomen. Each side could choose. And so underneath those dressings, a patient randomized to keyhole surgery, their incisions would have been placed here. And if they'd been randomized to a major incision, their incision would have been there. And those patients after surgery all had those same dressings put on. They were never told when they were randomized whether they'd have keyhole or open surgery. And then on day 2 post-surgery, the patient was asked to guess what operation they think they had received. And these patients absolutely couldn't tell what operation they had. And as we accumulated this data in the pilot phase, the surgical team began to realise and believe it that the patients just couldn't tell. The pain was similar whichever group they were in. And then the surgeon's equipoise grew. So it's really important, particularly in a pilot phase of a surgical trial, if possible, to test whether blinding of patients is feasible and it works. The next thing is it of course possible to ever blind the surgeons to the operation that they have conducted? I mean, it's a really good question, because the problem with having caregivers that aren't blinded, as you all know, is that you may get systematic differences between how the surgeons operate, between how they care for patients, which will lead to performance bias. And performance bias is a major challenge in surgical trials. You can't blind surgeons. We took this photo over 10 years ago now, that's me on screen right. For about 10 seconds, we had those blindfolds on while the photo was taken. It was very scary. So how do you manage performance bias in surgical trials? Well, what we're proposing is that we're developing methods, and Natalie Blencoe in Bristol has really led on this. We're developing methods that allow quality assurance of surgery to be agreed and to be conducted in the trial. And the quality assurance is dependent upon the overall design of the trial. The degree of quality assurance is dependent on the overall design of the trial. Because it's not possible in a surgical trial for each operation to be like each drug in a pharmaceutical trial. It's just not possible to standardize things to this degree. Surgical interventions are complex. In fact, they could be called complex interventions. Multiple components, operator expertise, the context in which they're delivered makes a difference to outcome. All these different factors, and the ones that happen before surgery and the care immediately after surgery, the pre, peri and post-operative care, all influences the patient outcome. So how on earth do you design interventions in surgical trials to account for this degree of complexity? And it really matters, because if you don't deal with it, then the results of the trials are questioned by other surgeons. They question what interventions were delivered, what was actually delivered. And basically, they criticise trials. We criticise trials and we say, if I'd done it my way, it's a sort of Frank Sinatra thing. If I'd done it my way, it would have been different. If I'd done it my way. If they'd had a local, not a general anaesthetic, the outcomes would have been different. If they'd used a metal, not a plastic valve, it would have been different. If 3 stitches, not 2 stitches had been used, the outcomes would have been different. So how do we design these trials? And it depends on how pragmatic the trial is. You know, there can be really pragmatic surgical trials and really tightly controlled ones. And the protocol for those 2 ends of the spectrum will vary. So in a very pragmatic surgical trial, the most pragmatic trial I've ever seen, the protocol said perform an operation, and it named the operation. Perform a carotid endarterectomy. It didn't give any other recommendations. And in the CRF it said what date was the carotid endarterectomy performed. No other information. At the other end of the spectrum, in a very detailed explanatory study, there'll be a very tightly controlled description of the operation that is to be performed. There'll be careful control of the person who is going to perform it, their expertise, and the co-interventions before, during and after surgery will also be controlled. And all of this will be very complicated and recorded in detail in the CRF. The 2 extremes. What is a way forward that we can use in surgical trials? Because they can both be criticised. Pragmatic trials are criticised for being sloppy, if I'd done it my way, explanatory trials are criticised because it's just not possible to repeat those things to that level of detail everywhere else. So what we need to do in the protocol, in the conduct of the study, is to agree on what's going to be done with some boundaries and flexibility, and then to monitor what actually is done and how it is done. So we have been developing methods for quality assurance of surgical interventions in trials, and there are 4 different components at the moment. There is information about the entry criteria. There could be information about the actual protocol, what is to be done. We would recommend doing all this in a pilot phase. Then we've developed methods for monitoring what's done during the trial with feedback to maintain the standards. And then it's possible to really try and look at whether the quality assurance, the key components of the intervention that is delivered, can be validated against clinical outcomes. I'm going to give you an example of how we've done this in a trial we've completed called the Romeo study, which has randomized patients with cancer of the gullet to keyhole surgery or to open surgery, the same trial in which we did the blinding study of patients. We did establish some entry criteria, but not every trial would need it. We said that every trust had to have done at least 50, to be doing at least 50 cancer resections per year, and that we would only allow them to enter the study if a minimum of 2 surgeons from each centre were able to participate. And before they could enter the study, we required each surgeon to send 2 videos of the operation in, and those videos were assessed. We kept all this on the delegation log. We made sure the videos were transferred and uploaded to Bristol. They were anonymised, and then the chief investigator, one of my surgical colleagues, and another assessor analysed every video. And they analysed every video for 2 things: for how the operation was done, the quality of surgery, and for what was done. And we used validated methods for doing that: quality of surgery and what was done. Now, the quality of surgery can be assessed with what's called an OSATS tool, and it grades it between 1 and 5. And they're the sort of things that, is the surgeon during the video showing respect for tissue? Are they making unnecessary movements, or are they making consistent movements? So we analysed them using this validated tool. And we also set a standard. We said that we expected that all the surgeons in this fairly pragmatic trial would be expected to perform at 3 or above. And then we looked to see if they could complete each task. And so for removal of the lymph nodes there, the abdominal lymphadenectomy, we looked to see whether it was complete. And you can establish whether it's complete by looking to see if those structures are visible, whether it was incomplete, or there was no evidence from the videos that whether it was performed at all, a so-called task analysis. That was the first thing we did. And then we, in the pilot phase, and once they had those videos assessed, they could take part in the trial. Now, in the main trial, in this study, we used a monitoring of what was done by taking photos in every operation and providing feedback. And the way we chose what matters in the photos is we deconstructed the surgical procedure to its component parts in the study team, and we tried to identify what mattered, which part of those operations we absolutely thought was essential to be done, mandated, which parts were optional, and which parts we thought should not be done in the context of this trial. And within the mandated components, there was some flexibility with boundaries. So depending on the overall pragmatic nature of the trial, this is the key part. Some bits would be very strictly mandated with a little flexibility in a very tightly controlled trial, but in a very pragmatic trial, there would be lots of things that would be optional. So this was all pre-agreed by the study team in the protocol, with input from the different surgical components. So just to go over that again, we split the operation, deconstructed it, discussed what could be delivered, whether it was essential, optional, or prohibited. This was fairly pragmatic, so we really only focused on certain things that were mandated. We made a lot of it optional, and we prohibited a few things. And then in the manual, we wrote what was required in the study protocol, and we monitored it. And you might historically have monitored this in the CRF, but what was written down can never be quite certain. So we monitored it in the CRF, but we also took photographs. We took some images of what actually had been done to make sure that what had been said had been done was actually done. And we had these agreed photographs for every procedure, and they were all uploaded remotely via a secure NHS portal. And we employed a research photographer who monitored all this and checked the images were accessible, and then the assessments could be made. The way we assessed, we had 7 images that we thought were important, listed here. And we did this kind of the whole way through the trial. And now, having finished it, I think we would reduce the number of required images, and we would set that all out in the pilot phase. I'm just going to show you a photograph of one. Sorry for those of you who don't like blood. This is just an image showing here that vertical structure in the middle is an artery. And the question was the removal of the tissue around the artery complete, incomplete, or not performed? And you can see an artery going vertically up and one on the screen left, and there should be one on the screen right. So this would be classified as an incomplete dissection. And that was, we tested that with our reviewers, and there was good. So we could tell whether it's accessible, unable to access, performed, or incomplete. And in fact, we recruited reviewers using a Twitter method, and we had 12 surgeons who assessed a lot of photographs, because you'll see in a minute how many. And we used multiple assessors of each image. And the final thing we did in the Romeo study was we used some specific clinical outcomes to try and validate our QA methods, which I'll come back to at the end. So a few results. There were 9 centres, so we had 27 surgeons submitting 2 videos each. That meant that we expected 54 videos. We actually received 60. And here's just an example of some of the feedback. So with 60 videos, grading the quality of surgery across the country, nearly all of those aspects were very well performed. There were some limitations with time and motion. And then when we did our centre feedback, we compared the centre scores with those for the whole group. And this is really interesting, giving centres their feedback. Surgeons have never had this level of detail on their surgery given to them ever before. When we looked at the task analysis, how complete were the different aspects of the procedure, this just shows you for the whole group that actually the proportions of complete on the screen right there were some parts of the operation that were incomplete across the board. And we then again gave the local data to compare it to the real-time data. And as in Romeo, we did it all at the end. In future trials, we'd do it as we go along to try and influence standards of surgery. And all the photographs, we had lots of photographs in. There were 310 patients randomized into this part of the trial. We expected of those, nearly all of them, 89%, had some sort of imaging performed, which meant we expected nearly 2,000 images. We actually received nearly 3,000 because people seemed very keen at sending in extra images, and also some unnecessary images of the window or their feet or something. A quarter of them showed the external wounds, which really mattered in Romeo, and 3/4 of them were intraoperative images. Then we had reasons for the necessary and unnecessary reviews shown here. And then just to briefly show you how these images did in terms of whether the aspect of the operation was complete or incomplete. And the bit that matters is that the bit in light blue, which is the complete dissection. So the band across the middle showed that complete dissection was performed in those areas, but an incomplete dissection was the yellow one, and the dissection was not performed, or sorry, I'm talking nonsense. So the dissection was incomplete or complete in the yellow and blue ones. So actually there was some areas of need for improvement here, or the photo was not accessible. And finally, we tried to validate some of these quality assurance measures by looking at the nodal count, the lymph node count per esophagectomy, which is an indicator of outcome, an indicator of cancer resection. And so we had some information about the extent of lymphadenectomy, which we could in future use to compare that with the completeness of the surgery. And we haven't done this analysis, but as time goes on, we'll be able to make an assessment between all the photos and the completeness of the lymphadenectomy with how many nodes were removed per case. And there's a lot of interest when we take this to the centres. The surgeons are very keen to see how their centre and their individual performance compared with the overall group. So this just shows you the different aspects of work that we've done in working out how to measure quality assurance in surgical trials. At the moment, there is 4 components. They don't all need to be performed in every study. We would recommend that these things are done in the pilot phase so that in the main trial, the videos or the photographs can be used in real time to monitor performance in the main trial and give feedback to surgeons during the main trial. And then at the end of the trial, we would then recommend doing an analysis to check that the standard of surgery performed is the same, to the same degree in both groups. And then that will give some evidence to show there is minimal performance bias. So to go back to Richard Horton, he said in 1996 that surgical research was like comic opera. That has changed. The number of surgical trials has massively increased. The culture is changing and the quality of the trials themselves is absolutely improving. I'm really pleased to have been part of this initiative. And what was once thought to be comic opera is now some sort of beautiful symphonic sound. And I just have started listing all the many people that have been involved in the work that I've been involved with. And this is an incomplete list now, but I want to acknowledge them all and thank you to them and the funders of all this research. Thank you for listening, and I'm pleased to take any questions.